Hello, my name is Matthew McLean. I'm a professor at the Masters University in California, and I'm also a vertebrate paleontologist and a creationist. And I'm going to talk to you today about synapsids and the evolution of mammals, a different view. Obviously, because I don't think that all mammals share a single common ancestor, and we're going to get into what that is. But your first question might be, what is a synapsid? Well, Synapsida is the group that contains mammals and their closest fossil relatives. So you can see over here we have a possum and a platypus, both being mammals we recognize, but there's all these other animals that are similar to modern mammals in many ways, but are not truly mammals. So we're going to take a look at some of these animals that are thought to be similar to mammals, but not quite. So you can see there are pelycosaurs, which are up here near the top where it says Dimetrodon. There are therapsids, which are these animals in here. And of the therapsids, there are the cynodonts, which Proboscis and Thrinaxodon are both examples of those. And then there'd be Mammaliaformes, which would be the group that contains things like Morganucodon, as well as our true mammals are in there. And so I should go ahead and say this. Technically, synapsid and therapsid, if you're using these terms as they're intended, they're going to include mammals as part of them. Now, you technically, if you want to talk about the animals, like for instance up here, that aren't mammals, you'd have to call them non-mammalian therapsids or non-mammalian synapsids, and that's very long. And so just for our purposes, for this lecture, I'll talk about when I say therapsids, I'm not including mammals unless I specify. So let's take a look at this therapsid line to mammals. You have your pelycosaurs, and then you hit true therapsida, and that includes animals like Pyramnosuchians, Dinocephalians, Anomodonts, Gorgonopsians, Theracephalians, and Cynodonts. And then, of course, the mammals at the end there. And so this is supposedly the evolutionary story for the transition from pelycosaurs to mammals, ultimately through therapsids. You can see there's a variety of animals represented here, they're really interesting. Take a little bit of a look at some of the other ones. Start with our pelycosaurs. These are the more reptile-like things. So you've got Cotylarhynchus here, the caseid. We've got this big barrel chest, really tiny head, absurdly tiny head. You can see it's a big animal. You could ride that thing. But yeah, not incredibly mammal-like when you look at it. They tend to have sprawling postures. You can see there with the humerus and the femur both held out to the side, more like what you see in a lizard. You have animals, sphacodontid pelycosaurs like Dimetrodon or Secodontosaurus, which may have been able to have a little bit more of an upright posture. There's some debate about that. People are trying to look into that, but still overall not all that much like a mammal. But then we hit our therapsids, and they are incredible. I mean, just look at this guy. This is Moschops. What a guy. He's got this also a big barrel chest, not such a tiny head, actually a head that super thick bone on it for butting into things. Kind of a big chest animal. It's a really funny animal. We also have our Dicynodonts. These are, the way I like to explain this to students is that they're basically, imagine a pig with the head of an eagle and the tusks of a walrus. So yeah, it's got a beak. But it's also got tusks, and it's kind of a pig-like body. And they'd be sprawling to semi-erect in posture. We have our Gorgonopsians here, and you can see these guys have saber teeth, as you can see right there. And they are carnivores. The other two we have here are herbivores, and they probably would be top predators in their environments. And you can notice it looks a little bit like a dog or a wolf, hence Lycanops, referring to a wolf. And my personal favorite, Astemonasuchus. Look at this, a fantastic animal. It's so wild, just fascinating that God created something like this. And you're like, okay, how do they actually know what it looks like? Well, there's the skull for you. So yeah, we have a pretty good understanding what it looks like. In fact, Astemonasuchus is one of only two therapsids where we have skin impressions, non-mammalian ones, obviously. So Astemonasuchus, there are some impressions of skin around the head, and it seems to be kind of naked skin with some bumps on it, maybe even some evidence of some kind of gland. Just recently, new remains came out of South Africa for what they're calling mummified Lystrosaurus. So Lystrosaurus is a Dicynodont, one of the ones with the beaks, and it has skin impressions all over the body, and seems to have that kind of similar naked kind of skin. Now, these animals I've shown you, most of them don't look all that much like mammals. And so you might think, why would an evolutionary scientist believe that therapsids form transitional sequences to mammals? Well, a lot of that goes back to the fact that I haven't shown you the more mammal-like ones. I've shown you the fun ones so far. But you can see like this artist rendition of a Gorgonopsian, a Therocephalian, and a Cynodont. Certainly they're making them look very mammalian. And so why would they be doing that? We'll look at these animals in a little more detail. But I want to first just give you a glimpse into how robust many evolutionists believe this transitional sequence is as proof for evolution. So here's a book called Evolution Slam Dunk: Why the Reptile-Mammal Transition Proves Macroevolution and How Anti-Evolutionists Ignore It. It's by James Downard. Obviously no friend to creationism. He writes, the reptile-mammal transition is the most compelling case for macroevolution. Besides the fossils covering millions of years, there's a wealth of corroborating science data from developmental biology to the latest genetics. Now, how do those skeptical of evolution, whether they call themselves creationists or favor intelligent design, deal with all that evidence? They don't. That's what he wrote. So like I said, not a friend of creationism, but you know what? He's pretty much right. For the most part, creationists have ignored these animals. There's a little bit of writing here and there in 70s, 80s, 90s, 2000s. You can find little articles, but it's really sparse that it's been dealt with at all. Now, I will say in the defense of creationists that evolutionists haven't always capitalized on this either. It's hard to find good popular books on synapsids that are even written by evolutionists. Most of this is hidden away in technical literature. So it's not like this is, just sitting out there for everybody to look at all the time and we have no answer. No, it's stuff that a lot of people don't know about regardless of their worldview. But I want to show you why is it that evolutionists are thinking of these animals as transitional. So we're going to take a look at the Gorgonopsians, which are, like I showed you earlier, a group of therapsids that have saber teeth. They are carnivores. And you notice, yeah, like I said, the skull does kind of resemble a wolf or something. Artist depictions of these animals, yeah, it looks like kind of like a reptile dog or something. Like you can imagine it like that. And what's really interesting about Gorgonopsians is they do show evidence for being able to switch between a semi-sprawling to an erect posture. So in the humerus and in the femur, you can see that there's degrees of rotation that are possible. And it has been talked about for decades that probably these animals could switch between the two when they wanted to. And as we make our way down that kind of line to mammals, we hit cynodonts. And cynodonts are the most mammal-like of all the non-mammalian synapsids. So like in mammals, the lower jaw of a cynodont is almost entirely made up of the dentary bone. So the other animals we've been looking at so far, they have multiple bones in the lower jaw. But in mammals, we only have one bone. It's called the dentary. It makes up our lower jaw. And so in cynodonts, you're seeing that right there. They also have a secondary palate, secondary bony palate. So this is something that mammals have that allows for us to be able to breathe through our nose while we're eating. And lots of animals don't have that. Lizards just have a big opening right there. They don't have a hard bony separation between the two chambers, but cynodonts do. Additionally, they have true lumbar vertebrae. So this Thrinaxodon right here, you can see a skull of Thrinaxodon. I'm also going to show you a video. So check this out. It's pretty cool. They did a synchrotron scan of this rock. It's a fossilized burrow, and inside of it, they found a Thrinaxodon cynodont with an amphibian called Brumastega. And you can see Thrinaxodon has some weird-looking ribs, but it does have a portion of the vertebral column that is consistent with what we see in mammals, the lumbar region, which allows for a lot of vertical flexion. It's like where you don't have ribs poking out from those vertebrae. And as we continue to look at cynodonts, we see even more mammal-like ones, like Probanognathus here, where if you look, almost the entire jaw is the dentary. So you can see almost the entire jaw is the dentary right there. But there are other bones. They're just way, way crammed back in here, kind of on the inside for the most part. And the jaw joint traditionally with reptiles is the quadrate and the articular. Okay, and that's what all of our cynodonts and synapsids we've looked at so far have had. But mammals have a jaw joint that's between the squamosal and the dentary. And what's really interesting is it looks like these are so close in Proboscis that it may have actually had two jaw joints, what we call a double jaw joint. It had one between the quadrate articular and one between the squamosal and the dentary. And this is very important because it looks transitional, right? It looks like you're switching from one type of jaw joint to another. And this was predicted before it was found. Paleontologists thinking about this said, well, if you're going to have a switch in jaw joints, you've got to add the second one before you get rid of the other one. And you should have some point where you have two. And here you've got an animal that looks like it has two. And then finally, we hit things that we would kind of call mammals if we saw them, certainly. There are some differences, though. That's why they're in this group Mammaliaformes and not truly within Mammalia. So their limbs are somewhat still sprawling, but they're pretty erect. And remember that, like a platypus and echidna have sprawling limbs. So that's not, doesn't make you not a mammal. They did have fur. How do we know? There are fossils like Spinalestes here where they have fur on them. And then also, they probably produced milk. And you say, well, how would we know that? Well, you can check at the way that they lose their teeth, and it seems to match what we see in modern mammals that produce milk. So this is good assumption, right? And it looks like a mammal, acts like a mammal, but there's some differences. For instance, if we look at the lower jaw of these animals, okay, like we're going to look at Ionoconodon, but also look at Morganucodon we saw on the last page there. Morganucodon, yes, its whole lower jaw is one bone, except there are some little bones on the inside of the back of the lower jaw, and they are holding in place where the eardrum would be. And you think that's kind of weird. Well, not exactly, because it turns out that our ears, okay, the bones around our ears, when we're an embryo, those bones actually come from the same embryonic tissue as our lower jaw area. And this was discovered before evolution was a widely accepted theory, before we knew about any of these fossil animals. It was discovered by an anatomist working on fetal pigs. And he realized there was a connection between the ear and the lower jaw. And so evolutionists taking that and looking at some fossils where you seem to have bones that look like they're supporting the ear and they're attached to the lower jaw, they're like, hey, that's what we predict as an evolutionist, right? Is the appearance of these things. And if you look at an animal like Ionoconodon, okay, you'll notice that you've clearly got your ear bones here, but they are still attached to the lower jaw by Meckel's cartilage. And you can see that, you can especially see this right here in this ventral view of the jaw, so the underneath view, that what's Meckel's cartilage right here attaches these ear bones to the lower jaw in we think, the adult animal. So Meckel's cartilage is typically something that only exists during embryonic development, and then it disappears. So here's an embryonic platypus lower jaw right here, and you can see the Meckel's cartilage, but in the adult, this disappears and these bones are no longer attached to the lower jaw, and that's an adult platypus ear bones right there. But in Ionoconodon, they're still attached. And so you can imagine here an evolutionist making a narrative with this, right? That the bones start in the lower jaw and our Permian synapsids. They get constantly pushed farther and farther back into the inside in our cynodonts. And then they turn into the ear bones. And if somebody told you, oh yeah, our inner ear bones, which are exquisitely designed, by the way, and are tiny, they're the only bones that don't grow your entire life, and they have this amazing articulation, all this kind of stuff. If somebody came and told you, like, oh yeah, those evolved from bones in your lower jaw and your ancestors, you're like, okay, that's really crazy and wild. But then when you see the anatomical and embryological data combined with the fossil data and the fact that these cynodonts are basically in stratigraphic order with the becoming more mammal-like, yeah, you can see how somebody would come to that conclusion if they were just looking at these evidences, not including, the Bible as evidence. If they're just thinking about these things, they'd say, yeah, no, it looks like our ear bones as mammals came from the jaws of these animals. And so just to reiterate this, your basal cynodonts and here, things like Procynosuchus, you can still clearly see the other bones at the back of the lower jaw. By the time you get Thraxodon, they're pushed way back there. When you get to Probanognathus, they're barely visible on the outside, and you still have that little jaw joint back there. By the time you get to Moroneucodon, I mean, they're way inside there. And they may still function partially with the jaw. It's a little bit unclear, but I mean, it looks essentially mammalian at that point. And so these are in stratigraphic order, right? These guys show up first in the fossil record, then Probanognathus, and then Moroneucodon. So what do we do at this point as creationists? Like, do we just admit defeat? We're like, okay, I guess you guys win. Well, no, we know the Bible's true, right? That's not up for grabs. I mean, the Bible's very clear that God created different kinds of animals. And so we can't have all mammals, including you and me, descended from a single common ancestor in the synapsids. I mean, that doesn't make any sense. That doesn't fit with what the scriptures teach at all. So, well, we could just ignore this stuff or try and deny that the fossils are real or something, but we've been collecting these fossils for over a century. I mean, many of these things have been known for a long time. This is not, there's no reason we should doubt these evidences. So what can we do? Well, this is where creationists want to step in and do research, right? I'm a young earth creationist. That's means that I think the account given in scripture for the origins of the universe and all it contains is historical. And as we said, part of that account describes the creation of separate groups of animals, kinds it refers to in the Bible. And so we don't think that all animals share a single common ancestor. And we know specifically for sure humans have their own origin, right? Whereas evolutionists are claiming that cynodonts are the ancestors not only for mammals, but also us being mammals. So yeah, I think that we have reasons to doubt that, we share a common ancestor with this guy. So what can we do? Well, this is where I started working with some of my students at the Masters University to try and locate, first of all, can we figure out created kinds, right? So if the Bible is true, which it is, that God created different kinds of animals during the creation week, so they have separate origins, they don't share a single common ancestor, we should be able to find evidence for that, we would think. And so we decided to start doing this kind of work on created kinds with synapsids. And we published our results in a series of abstracts in the Journal of Creation, Theology, and Science because we were presenting them at the Origins Conference. And so we did several different abstracts looking at these animals. And we were presenting these at sequential Origins conferences, and you can look these up. They're free online, the text of these abstracts. The Origins Conference is the annual meeting of the Creation Biology Society, Creation Theology Society, and Creation Geology Society. And it's a really cool conference if you're ever able to go in the United States. It's well worth your time. It's always in July. And this year we're meeting with the International Conference on Creationism, kind of a joint meeting. It's a cool opportunity, and we were able to look at it. So let me show you just a little glimpse at what we did. We did what's called baraminology, and baraminology is the study of created kinds. And with the study of created kinds, we're scientists, right? We don't just want to subjectively be like, well, that kind of looks like that, so yeah, they're in the same kind, but oh no, that looks really different from that. No, we want to find a way to make it a little more objective, right? And so that's where statistical baraminological methods came in. We were taking a big data set full of information about different species, and you're putting it into a computer program, and it's telling you statistical information on what is significantly similar or dissimilar. And so the hope is that animals that are really similar to each other, they're going to group together, and animals that are very dissimilar, we should see evidence of what we call discontinuity, separating them from other animals. And so when we look at the cynodonts, lo and behold, they are not one giant continuous series, right? If this, and I know you may not be familiar with looking at these kinds of plots, but if it really were just one giant line, right? One giant flow of less mammal-like to more mammal-like to more mammal-like to mammals, then we would expect to see a giant stream of black squares here going up the middle of this diagram, okay? we would not expect to see discrete units. And that's exactly what we see, is discrete units, which is what a creationist would want to see. It's what we would predict, is that there should be distinct created kinds. And so these blocks you can see here are very separate from each other. There's shared information between these two blocks, but these two are very separate. And so this is not a continuous series. And remember, these are the cynodonts. These are the ones that are supposed to be the most mammal-like, that have the best transition, the crown jewel of evolution. And it's not a continuous series. You can see it easily punctuated with discontinuity. And that's exciting. And when we look at our multidimensional scaling results, we see the same thing. We can see very different groups separated from each other. Now, like I said, the blue and the green here, they're close together. Maybe that is one created kind. Maybe it takes more work to separate it out. I don't know. But we can clearly see discontinuity between those and our red group here, and then also with our yellow group. And that's cool, because there's no reason it had to fall out this way when we put the methods in. We put the data in there, we ran it. It could have just been a single stream, and we'd have to think through, what does that mean? But it wasn't. It was falling into discrete groups, and that's really exciting. And this didn't just happen for cynodonts, it happened for every one of the therapsid groups we analyzed. And this isn't entirely unsurprising, actually, because even in the conventional literature among the evolutionists, these discussions about very distinct body plans of therapsids are common. So here's some figures from an article by T.S. Kemp, who's one of the experts on therapsids from back in 2009. And you can see he shows an ancestral therapsid, which is essentially a Bjarmasuchian, same kind of look. Dinocephalians, Nomodonts, Gorgonopsians, Theracephalians, Cynodonts are 6 main types of therapsids. And he says they're all so different from each other that I can't imagine one of them turning into another one. Like, they all are set up very differently in their skull constructions and jaw attachment for muscles. And not only that, but they show up essentially simultaneously in the rock record, both in Russia and South Africa. Yes, cynodonts show up later than the other ones, but all the other groups essentially show up at exactly the same time, fully formed. And he said it's really interesting. In the article, he actually says it's a lot like the Cambrian explosion, or the sudden appearance of mammals and birds in the Paleogene. I'm reading that and I'm like, wow, like that's what I would say as a creationist, is it looks like they just suddenly appear in the rock record, fully formed, without ancestry, and they're distinct. And that's cool. Like, that's very much in favor of what we would think. And so I showed you earlier this flow from a more pelycosaur-like animal through the different therapsids all the way to mammals. But what we found as creationists was that this did not exist. In fact, every one of these groups was discontinuous with the others. Not only that, but several of these groups were actually made of multiple created kinds. And so this is not just one big continuation of evolutionary series of crown jewel of evolution. No, in fact, it's actually punctuated by these different individual created kinds. And that's cool. That's exciting. We were thrilled about that. But we still have a lot of questions. For instance, why do they look transitional? I mean, yes, it's cool that there's separate created kinds and we can find discontinuity and see that, but why does one look kind of more like a mammal than another one? Okay. Well, I think that some of this has to do with our misconceptions of how nature is organized. Okay. So when you think about our living types of vertebrate animals, right, we have birds, we have mammals, reptiles, amphibians, and fish. And they're all very easy to tell apart, right? No one ever looks at a fish and is like, is that a mammal? Should I keep that as a pet in my house? Like, no one ever looks at a bird and says, I wonder if that's actually a reptile. Like, they're very, very distinct groups of animals. And we named these groups based on, the distinctions we see today around us. Then we started to find fossil creatures, and many of those fossil creatures readily fit in our groups, right? Woolly mammoths and saber-toothed cats are obviously mammals. But other animals in the fossil record didn't fit into these groups. And so our temptation a lot of times is to just shoehorn them in, just fit them in and be like, no. Like, these ones are birds and these ones are reptiles and these ones are mammals, and we just do that. We try and fit them into those groups. But that's not really being fair to the patterns that we see in nature. It would be much better if instead we tried to create our classification based on what's actually out there, right? What's the pattern that God used in designing things? And that's what we want to do. And so, yes, an evolutionist can look at something like this and see a phylogeny, see an evolutionary tree, right? They move from fish to amphibians to reptiles and splits up into two groups, one going to mammals, one going to birds. But you could also choose to look at this and see a larger design picture at work than we previously knew, which is exciting. At this zoomed out scale, the divisions between these groups are not as clear as they used to be. But is that really that unexpected? I mean, life is full of continuity and discontinuity, right? You look a lot more like a monkey or a pig or a dog than you do a jellyfish or a tree. I mean, it's okay to say that. There's nothing wrong with that. And yeah, if I had a data set where it was, all mammals and then one tree, guess what? All those mammals are going to look super, super similar than they are to that one tree. And so you got to look at the right scale. Okay. So let me help you a little bit understand this with analogy with mammals. Okay. So we all recognize that mammal is a group. It's a word we use. It talks about animals that, have fur, hair, they produce milk, they usually give birth to live young. Now, most people, when they say a bat and a whale are both mammals, they just mean that, they have the same traits, right? They both produce milk and stuff. But when an evolutionist says that a bat and a whale are both mammals, what they actually mean is that they share a common ancestor with the rest of the mammals more closely than the common ancestor with, reptiles or birds or anything else like that, okay? So we can add in here as well, we got some wombats and red pandas, right? And it's really easy for us to tell these apart, okay? Now, we'd recognize all these as mammals taxonomically, but we'd easily recognize these are not the same created kind, right? Like, I don't think that all four of these animals share a single common ancestor. I mean, that'd be kind of wild. I can easily tell apart created kinds, or at least something like created kinds. And even when we add fossil mammals, it doesn't really change that picture. And the same is true when we add in our synapsids. We can still see different kinds. Yes, can you see something that's the most mammal-like versus the least mammal-like? Sure, you can make those kinds of designations, absolutely. But I can still see the created kinds. Acknowledging the taxonomy doesn't automatically stop me from being able to pinpoint the different created kinds. Now you might be saying, okay, that's great that you were able to pinpoint created kinds, potential created kinds anyway, among the therapsids, but what about all the other things, right? I mean, why are they in the stratigraphic order that they are, right? I showed you earlier that the most mammal-like of the cynodonts are the highest in the fossil record. And as you go lower, they look less and less mammal-like. So what does that mean? Well, we attempted to answer that in another presentation at Origins in the Journal of Creation, Theology, and Science. You can read about that. It's in the 2019 issue. And we still don't really know entirely. We've gotten some hints here and there. There's some good things to look at. And I encourage you to check out that abstract. And it's definitely an area we're still working on. But that's not the only thing we need to figure out, right? I mean, what about that Joddy ear evolution thing? If that has nothing to do with the evolution of ears, then what does it mean? I think that's a great question, and I want to start exploring that. But it's going to require a lot of work because you're going to have to dive into embryological research. You're also going to have to dive into genetics research, all kinds of things that I'm not qualified to talk about. And so I'm going to have to collaborate with people, which could be really cool. And I'm excited about the possibilities there. But one thing we can look at for the rest of our time today is the rest of the skeleton increasing mammalness, right? So there's this idea that when you go back to pelycosaurs and some of your first therapsids, they're more reptile-like in the postcranial skeleton, but as you get closer and closer to mammals, that it's not just the jaws and ears that are changing, it's not just the skull that's changing, but the rest of the body's changing as well. And one particular area we can look at is fingers and toes. And you say, why fingers and toes? Okay, well, I'll show you some hand skeletal diagrams here. We have one on the left, one on the right. Okay, you'll notice that the one on the left has, we can count the number of finger bones, of phalanges here. So you got 2, 3, 4, 5, 3. Who has a hand like this? Why, many reptiles, like the Tuatara. But this other hand here goes 2, 3, 3, 3, 3. And you might be sitting there counting your own finger bones at this point and realize, hey, that's my hand. Yes, mammals have the 2, 3, 3, 3, 3 situation. So let me show you some other hands here. The first hand here, letter C, is from Dimetrodon. And you can see it goes 2, 3, 4, 5, 3, which is what you would expect, being kind of a reptile-looking animal. The next hand here is also 2, 3, 4, 5, 3. But notice there's something strange going on. Like, look at these little phalanges in gray. These little finger bones in gray. They're like tiny little discs. In fact, sometimes they're actually fused to one of the other finger bones, which is kind of weird. And that's from a therapsid. It's from a Gorgonopsian. So what is going on here? Well, the evolution narrative is that these little finger bones are on their way out, right? So these animals start with the reptile-like hands, and then as they become more mammal-like, they have this more erect posture, they're doing more things, they're more active, those little finger bones start to disappear. And that's how they do it, is that they turn these little discs and then they fuse to another finger and just kind of, another finger bone, sorry, and they turn into the 2, 3, 3, 3, 3 situation. So that's what we're going to be investigating. And if we look at the hands from our different therapsid groups, you can see there all 6 groups. The mammalian condition shows up in 5 of these groups. So the 2, 3, 3, 3, 3 condition is in 5 different groups. It's these groups: Dinocephalians, Anomodonts, Gorgonopsians, Therocephalians, and Cynodonts. But there's actually a problem here. And I'll show it to you by going back to our chart from before. Shows up in these groups. Okay? And you think, okay, well, why is that a problem? Because it's not the only condition in these groups. I just showed you a minute ago a Gorgonopsian that had 2, 3, 4, 5, 3 finger bone combination. So you're telling me some Gorgonopsians also have 2, 3, 3, 3, 3? Yes. And these groups, some of the members have the mammalian condition and some of them don't. So how do we explain that? That's kind of weird. So one potential solution is that most or all of the therapsids had the mammalian condition, but some of them reverted to a reptile or reptile-like condition. Okay, and so this was proposed by Rowan van der Heever in 1986. And so you can see everybody starting with the mammalian condition down here in the phylogeny, and as you go along, some of them revert back to the reptilian condition, which was the actually plesiomorphic ancestral condition way back. So that was one idea. It's a little bit of a strange idea, for sure, that these animals are all kind of reverting. Some of them are reverting back, some of them are not. It's kind of unusual. An alternative was that mammalian or near mammalian condition evolved multiple times. And so in this model, you can see you start with the reptilian one, and then the mammalian condition evolves in Dinocephalians, it evolves in Anomodonts, it evolves in Gorgonopsians, it evolves in Therocephalians, it evolves in Cynodonts. Easy, right? Kind of what it seems like. And so that was Hobson's suggestion. Okay, and so for instance, new data has come in. So you see Anomodontia is only 2, 3, 3, 3. We actually now have a basal anomodont, or supposedly a primitive anomodont, that is has the reptilian condition in the hands. So that does seem to fit Hobson's idea a little bit better than the one that was suggested by Rowan van der Heever. What's really interesting, this paper where Hobson suggested this came out in 1995. And no one, as far as I can tell, has ever attempted to reanalyze this or challenge it. It's just kind of accepted as like, well, that's must be how it is. That's really interesting. Like even recent publications that get into fingers and toes and things, they just cite Hobson 1995 as the authority for explaining the trends of digit evolution in the synapsids. Additionally, the way he came up with this hypothesis, he tested it with phylogeny, okay? But no one, as far as I can tell, has ever tried to investigate these digit formulae with stratigraphy. So what do I mean by that? Okay, what if we compared the different digit formula possibilities to order in the rock record? So here's the geologic column. Okay, we find our non-mammalian synapsids in layers from the Carboniferous up to the Cretaceous. Most of them are Permian and Triassic. Okay, so in the Carboniferous and the Permian, you have your pelycosaurs. The therapsids show up in the Permian, go up into the Triassic, and then cynodonts continue on past the Triassic into the Cretaceous. And of course, mammals are supposed to evolve in the Triassic, Jurassic, and go onward. And from a conventional perspective, we would expect the reptilian condition should appear first and then the mammalian condition later. And in general, you see that pattern, right? You have pelycosaurs, then therapsids, then mammals. But we want to get down in the details, the nitty gritty. We want to understand what's going on here. And you might be like, well, how valid is this? Like, can we really be that certain about their position in the layers? Well, it turns out a lot of these therapsids come from places that have been studied for a very long time stratigraphically, like the Karoo Basin, for instance, where they have incredibly detailed stratigraphy for thousands and thousands of specimens. So yeah, that's pretty cool, and we can look at that and investigate it. So in order to do that, what we did, and I worked with a student at Masters here named Emily, we collected digit formulae for hands and feet for as many synapsids as possible. You can see I listed them out there if you're curious. We collected stratigraphic data on those taxa from something called the Paleobiology Database, which has tons and tons of information that's been collected and put up online for free. And then we try to find a way to graphically represent the data. I'm not sure we found the best way to do it right now. This is still a work in progress, but you're getting a little glimpse at it, which is fun. And so you can see here, we've got our different states. So this is the reptilian state, 2, 3, 4, 5, 3. This is the mammalian one right here, 2, 3, 3, 3, 3. This is just a weird other state that exists. And these other ones are other possibilities that some of our therapsids show. Okay, so when you see a black border around a dot here, that's indicating certainty. When it's not, then we're not so sure about it. And you can see the colors down there are our different synapsid groups. What's really, really interesting here, notice the mammalian condition 2, 3, 3, 3, 3 is present in 3 of our groups from their first appearance in the fossil record. Like, that's really interesting and unexpected, right? Our only group that really shows a transition from 2, 3, 4, 5, 3 to 2, 3, 3, 3, 3 with stratigraphy, you might argue for Gorgonopsians, but I think that's more an artifact of preservation than anything else. Really, cynodonts are the only ones. So cynodonts here are this kind of like turquoise teal kind of color, and you can see that they start here at 2, 3, 4, 4, 3, and then move into 2, 3, 3, 3, 3 later on. Here it is for feet, so looking at the toes, and we see the exact same pattern. The first Dinocephalians, Therocephalians, and Nomodonts you have in the fossil record, they already had the mammalian condition. Even though some Dinocephalians didn't, right? Some of our Nomodonts have a different condition, and they're actually found currently higher up a little stratigraphically. So we're not seeing the pattern. The only one, once again, we're seeing the pattern with is the cynodonts that we see at transition here from 2, 3, 4, 4, 3 to 2, 3, 3, 3, 3. And by the way, I didn't mention this before, but actually toes are a little bit different than fingers in reptiles. So the fingers is 2, 3, 4, 5, 3, but in toes is 2, 3, 4, 5, 4 instead. Okay, so what trends do we observe here? Mammalian formula appears in multiple groups at the same interval as the reptilian formula in therapsids. So from the moment we have therapsids in the fossil record, you already have some with mammalian condition, like Dinocephalians and Nomodonts and Therocephalians. Dinocephalians also show multiple strategies, as probably Anomodonts do too, right away. And that's really interesting, right? Unusual kind of intermediate digit formulae sometimes show up after the mammalian condition, which is surprising too. Only the cynodonts really show something that looks like or could be interpreted as a transition from an intermediate condition to a mammalian condition that actually works stratigraphically, which is really fascinating because that's not what I would have expected from what you hear talked about and what's actually written in the literature. You'd think it would be this nice like reptile to mammal kind of transition and the fingers and toes, and it's not. So what does this actually mean? We don't see any evidence for a general progression from more reptile-like to more mammal-like fingers and toes. That's really cool. because it means we don't have any particular reason to agree with the evolutionary narrative here. Yes, there are some synapsids with mammal-like digits and some have reptile-like digits, but they don't automatically agree with the evolutionary story. That's great. But listen, we can't stop there. It's not enough to say the evolutionary explanation is insufficient. Boom, I'm done. No, as a scientist, I want to know what it does mean, right? Because there's some weird stuff going on here. Like some groups have like more than one condition present. And it'd be really nice if like each different finger and toe combination was like a different created kind, but I just don't think that's the case. I mean, like look at Gorgonopsians, for instance, right? So in Gorgonopsians, we know of 2 different conditions in the hand, 2, 3, 4, 5, 3, and 2, 3, 3, 3, 3. So they either have reptilian or mammalian. Now, Gorgonopsia as a whole doesn't really have that much variability. Like the animals, they can be small or they can be big, but they all kind of look like the same kind of thing. And sure enough, when you run the baromenological analyses, I mean, yeah, there might be some evidence of discontinuity, but these could easily just be monobrahmins too. When you look at the different finger states, digit states, okay, our one specimen that shows definitively 23333 is Arctognathus right here, Arctognathus curvimola. He's not in a weird spot baromenologically. He's right there grouped with Lycanops. Shows positive correlation with him, okay? And he clusters with them over here. So yeah, it looks like we have a created kind right here where you've got both states present. And I think we can find in other examples too, where it looks like you have multiple digit formulae in the same created kind. So what does it mean then? Well, we have to say variability in digit bones is common across Therapsida. And we can ask why? Because we don't see that in mammals, right? We don't see this kind of, we see it, in a few different weird things like dolphins or something where they're making like a flipper, and that's really interesting. But like just normal hands and feet, we don't see this kind of variability that often. So why is this the case then? Well, could it be maybe developmental plasticity? So like not every member of the group grows the same way, and not every species within the created kind grows the same way. Maybe they respond differently to different stimuli in the environment, and it'll change based on that. So there's a study that was done a few years ago where they were taking some fish that can go on land a little bit, and they were raising them either in tanks with more water than land or more land than water. And it turns out that the fish's fins grew differently. The ones that had more land, their fins were actually stronger because they could go up on land more. So maybe there's something going on like that. Maybe it's related to some kind of locomotion difference that's in these animals. And so we were trying to think through these things, really racking our brains as to what's going on. And a new paper was published in Current Biology where they looked at the evolution of the reptilian gait into the mammalian gait, right? So they're saying, okay, where did mammalian locomotion come from? It had to come from reptiles in their evolutionary model, reptile-like creatures anyway. So they said, well, we predict that, the reptile-like gait must have evolved into the mammalian one through some kind of transition. So reptiles, as they walk, their body is curved side to side, okay? And mammals, as they walk, their spines kind of go up and down for the most part, right? Like you watch a cheetah run, like it looks like an accordion coming in and out, okay? So everyone assumed that non-mammalian synapsids would fit into some kind of position between those two, right? We're like a nice transitional state. They were surprised to discover, however, that they didn't. Because it turns out non-mammalian synapsids, for the most part, have very stiff spines. They don't bend all that much. And so they don't actually form a nice transition between, so over here on this plot, right, you see the green dots here are the reptilian ones, and the orange ones, and also some amphibians are pink here, and then the orange ones are mammals. And so you'd expect your non-mammalian synapsids to be right here, right in between these two, and they're not. They're way up here. So instead of forming like a nice transitional state between the two, they're just off doing their own thing, which is great, right? As a creationist, I'm like, that's exciting. But how does this help us with our problem? Well, it's interesting having these stiff spines. Having a stiff spine might allow for this variability to occur, whereas these other possibilities, it needs to be more restricted. At this stage, we don't really know, but it's really interesting that they're doing this kind of unusual thing with their fingers and toes, and they're also doing something unusual with their spine. And so really, we're just beginning to understand these animals even for the evolutionists. And so it's exciting for us as creationists to be able to jump in there and do some more research. And so, like I said, this finger and toe research is still in its preliminary stages. We still haven't published this, but we are excited about it and looking forward to seeing where it's going to go. And as part of this Therapsid research, I'm working on a big work that will cover creationist perspective on these groups and get the ball rolling on this kind of research. And hopefully we'll get more and more students and more collaboration happening to get a better understanding of these animals. So let me wrap it all up for us, okay? Give you a few points to kind of take home. If you fell asleep, here's your chance to wake up. Number one, there are different created kinds of what we call mammal-like reptiles or non-mammalian synapsids. It's not just one big continuum. It's actually discrete created kinds, which is what we would predict as a creationist, and that's cool. We see evidence for that, and that's exciting. Secondly, we don't need to be scared of animals that look transitional. Okay, I think it's many times as creationists, we see these things and we're like, oh, it looks too much like evolution. I don't want to deal with that. No, these are God's creatures, and we should try to understand them, and it's exciting to try and understand them. And listen, we don't need to be scared of scientific discoveries. There's cool stuff happening out there, and evolutionists do good research. Yes, they have a worldview that is incorrect, and it can taint things in their thinking and their interpretations, but they discover cool things, right? This is an image from a paper where they looked at a cynodont named Cantotherium. They found a specimen that had a mother and 38 babies with it. 38. That's a lot of babies, right? That's interesting. They're suddenly asking those questions like, what are they doing? They must be laying eggs. I mean, it would be really weird if they are giving birth to 38 babies at the same time. So there's lots of questions going on there. And listen, God's creations in the past, in the present, and one day in the future, hey, they will all declare his glory. And they will declare his glory for all time. And it's exciting to be a part of that process as a paleontologist, that I get to go expose myself to glory God and what he has made, and then I get to show it to others and help them also see God's glory as he intended it to be seen by us humans. Thank you for your time.